All right, welcome to the final podcast for day 39, chapter 9 on respiration. In this podcast, we want to talk about how a cell can generate energy in the form of ATP when oxygen is not present. And there are two pathways we want to talk about. And the first one is fermentation. We'll spend a little bit more time on this one than the second one, which is anaerobic respiration. You'll get much more of this when you take both of these when you take microbiology. Well, let's go ahead and get started. All right, up to this point, we've talked about cellular respiration in the presence of oxygen. And we talked about it as the importance of this because oxygen serves as a great final electron acceptor in the oxidative phosphorylation pathway. And without this, the cell dies and ultimately the organism could die. However, we know that there are some organisms, like our anaerobic bacteria, even some cells in eukaryotes, have to rely on alternative pathways because they also lack oxygen. Muscle tissues under high endurance are an example. So there are two pathways we can talk about, and we will briefly. The first one is anaerobic respiration. And the second one is fermentation. Let's begin with anaerobic respiration. I'm going to rewrite it up here, anaerobic respiration. I don't want to go into too much of detail with this, except to say that it's very similar to aerobic respiration, where we use the oxygen as the final electron acceptor. However, if you happen to be a bacterium that lives in an environment with no oxygen, what are you going to do? Well, one of the strategies they have developed is to use another chemical instead of oxygen as that final electron acceptor. A common one is sulfur. So sulfur is an alternative electron acceptor for bacteria. You may have experienced this yourself, not yourself, but you may have walked around areas where there have been bacteria that rely on sulfur as their electron acceptor, the final electron acceptor during anaerobic respiration, because it has this very bad odor of rotten eggs. If you ever smell that and you're out in the bouts, and you very well may be smelling the product of anaerobic respiration. You'll get more of this when you take microbiology, but for now, the main take-home point is that anaerobic bacteria can undergo anaerobic respiration by using an alternate final electron acceptor because there is no oxygen available for these particular bacteria. Next, I want to talk about fermentation. Fermentation is another pathway that can yield ATP with no oxygen. Well, now how can it do this? Well, a great example of this are yeast cells. Yeast cells can take glucose and convert it to pyruvate. Yeast cells can do two things with this pyruvate. They can have it undergo the pathway of aerobic respiration. In that case, it's going to take it through the citric acid cycle and through oxidative phosphorylation. Sorry about the abbreviations there. Just don't have enough room to write them all out. But this is what we talked about before on the last podcast. Alternatively, and important to remember, if this is the case, oxygen has to be present. However, if there's no oxygen present, or for some reason they are not using it, but let's just go ahead and say no oxygen. or it can be less oxygen. It might still choose this pathway. And it can go through a series of pathways to produce ethanol. So the question is, how are we making ATP in this pathway? Well, we make ATP in this pathway because glucose to pyruvate makes 2 ATP. And what this does, the reason why this pathway is important making ethanol, is that it takes the NADH from glycolysis, because NADH is a product of glycolysis, and it converts it to NAD+. NAD+ is essential to allow glycolysis to continue. If we didn't have a way, or the yeast cell didn't have a way to recycle and make more NAD+, it would not be able to continue to undergo glycolysis. So that's why this pathway, and I didn't call it before, but let's call it what it is now, this fermentation pathway is so important because it regenerates this NAD+ that can now be used to allow glycolysis to continue. Now granted, we're only making 2 ATP per glucose molecule, so it's not very efficient, but it's enough to keep the cell going. As a byproduct, it makes ethanol. Now, we as humans, we take advantage of the yeast to force them to undergo fermentation to make ethanol, and in this process we can make things like alcohol. Also through fermentation, we can make bread and other products. Other eukaryotes, even human cells, can undergo fermentation as well, not as a way for our whole bodies to survive, but in some tissues at certain times we can undergo fermentation. So in humans, we don't make ethanol, we make lactate. This is the chemical that builds up in our muscles when they're being overworked and makes our muscles cramp up, because those muscles tissues can no longer undergo aerobic respiration to make all that ATP, but they can ferment and keep the cells going with this minimal amount of ATP, but a byproduct, lactate or lactic acid, builds up in the muscles and can lead to things like muscle cramps. And we should also, just in case it pops up somewhere along the line, remind you that this all occurs, glycolysis, as you were told before, but also fermentation, this all occurs in the cytoplasm. The main things you should know from fermentation is that it takes pyruvate in an anaerobic pathway without oxygen and ferments it to generate ethanol. And it does this to allow glycolysis to continue. That's all we have for this third and final podcast for day 39. If you have any questions, please let us know. Thanks. Bye.